Hi, everyone. My name is Jordan Sicherman. I'm a second-year master's student in Dr. Paul Pavlidis' bioinformatics lab at the University of British Columbia. And today I'll be talking about my thesis work, which involves the large-scale mining with differential gene expression data with the goal of gaining insights into gene function. To first make sure everyone's on the same page about what this data is, I'll give a brief overview of differential expression analysis. Essentially, a researcher gathers gene expression data from two groups of biological interest. And so these two groups are meant to represent unique biological states that are being compared, typically a control group and a condition group. And this condition group may be a disease group, a treatment group for some drug, or et cetera. With this data, a statistical test is performed to examine which genes change in expression level across the comparison. This results in a ranking of genes by statistical significance that have a suspected involvement in this change in phenotype. After thresholding at some level of confidence, what results is a set or a hit list of genes whose expression level changes in this alteration of state. And we infer that that hit list is functionally involved in the change in phenotype. Whether these hit lists arise from differential expression analysis or GWAS or any number of other methods in genomics research, these hit lists are quite common and quite difficult to interpret. And that challenged interpretation arises because they are often large sets of genes that lack any amount of structure. Doing the wet lab follow-up on them is demanding and expensive and often intractable for large sets. So one way it's currently done is via Go emission analysis, which is a computational method that uses functional annotations to obtain a set of pathways or processes that are over-enriched in their list of genes. So this leverages the gene ontology, which is a structured set of annotations. And while useful, this analysis is still far from perfect. One notable shortcoming is that it depends on having functional annotations pre-assigned to genes, meaning that this method has a limited ability to make new discoveries. It's rather often used for a kind of sanity check for researchers to confirm that functional groups that they expected to see enriched in their gene set are in fact there. A different approach, given that we know that differential gene expression data encodes information about gene function, we in my lab have been exploring ways to use it at scale as a kind of complementary analysis to Go enrichment. This is enabled by public repositories such as the Gene Expression Omnibus, or GEO, containing a plethora of data. As you can see here, over 100,000 transcriptomic data sets are in their holdings. However, due to the nature of these repositories, the data inside of them is not in a format that is very permissive of this mining. There's a few reasons for this, but it essentially boils down to the fact that these repositories want to encourage users to deposit their data in mass. And so the formatting requirements are rather lenient, resulting in data that has been subjected to a variable amount of quality check with different pipelines and quite unstructured data resulting from it. Luckily, a big effort in my lab to date has been automatically pulling this data from public repositories. So you can see in the top right corner here that we source from a number of these public repositories, GEO, Sequence Read Archive, Array Express, et cetera. And beginning from the raw reads, all of the processing is done in-house through a standardized pipeline. And this ensures that there's consistent probe set mappings, consistent quality checking, and what have you. This gives us a large database of almost 11,000 studies, which covers a good fraction of GEO for human, mouse, and rat studies. It also has a significantly large or diverse set of condition comparisons in the holdings. As you can see, that's figured down here, over 60,000 unique comparisons inside of our database. And moreover, while there is this broad coverage, there's also a good amount of overlap between independent studies examining similar contrasts. And just to sort of repeat myself, these contrasts are condition comparisons. So comparing some control group to some condition group. The last thing I want to mention about this database that we use is that a team of undergraduate curators step in and do annotation on this database with labels from controlled vocabularies so that all the experiments have ontology terms, or most of them at least have ontology terms representing what the experiment is studying, making this mining very easy. Within this large corpus of differential expression analysis results, we have a tremendous wealth of information on gene function that's perfect for mining. And this is where my project centers. So you can imagine this corpus as sort of stacks of these gene by sample matrices for the differential expression analysis. And one way to mine it is by inputting a gene set that we have some sort of a priori interest in. In this case, we have a set of a few genes. Expression level was found to be activated by treatment with hydrocortisone. We can search across this entire network in some sort of automated way to find a consensus on which condition comparisons demonstrate high relatedness to the query. And that means that there's lots of differential expression and frequent differential expression for a given contrast. The general hope in this kind of analysis is that if you input genes that you have found that are involved in some specific response, you would hope that, well, A, those or similar conditions come out in the top few results for the enrichment to sort of validate your findings. So hydrocortisone is a glucocorticoid, and you can see here that a bunch of glucocorticoid drug treatments are showing up in the top results. But the other hope in this kind of analysis is that there are additional unexpected results that you can use to aid in the interpretation of your gene set. And so in this specific example, one could conclude that by inputting these genes that were found to be differentially regulated by treatment with hydrocortisone, they're not actually specific to hydrocortisone, but rather a general response to glucocorticoid treatment. If you're gathering new data and trying to interpret your hit list using this tool, you can also imagine finding an explanation for genes in your hit list that are unrelated to the condition you're studying. And so, for example, this ventricular assist device contrast showing up here, which is a treatment to control for heart failure. The authors of these studies found these genes to be dysregulated after this surgical procedure, but their study did not control for patients receiving treatment with glucocorticoids to manage the inflammation that occurs post-surgery. And so they could have used something like this to interpret their hit list and sort of deprioritize those genes as some potential confound. This kind of analysis is reminiscent of Go-Entry analysis, where you input a gene set and output some list of ranked labels. But since it does not require pre-assigned functional annotations and rather only depends on the count data, it can proceed in a more unbiased way. As I alluded to before, this hasn't been possible, at least not effectively so far to date, because the data has not been very permissive of it. And so at a high level, my work is sort of this middle step, and that is developing the algorithm to calculate these enrichments in a way that's meaningful and reliable. The algorithm has gone through a few iterations of development, but the overall bottom line is sort of as follows, and that is we want to capture meaningful features in the expression data while simultaneously minimizing the noise that's going to be inherent in this kind of corpus. So the process, just to sort of generalize it in a few steps, is roughly these 3 steps. And the first is to reduce the search space, or to reduce the experiments into these so-called gene components. And previous work in this area has so far used gene signatures as some sort of binary process where it's either differentially expressed or it's not differentially expressed. But we find that this is too low resolution view on the problem. Instead, we incorporate many per gene features, such as how much the expression level changed, how frequently that gene changed expression level across the entire corpus, and a few more, I think up to 6 other features we incorporate. And then we perform a bootstrap of 500,000 queries of random gene sets to create an empirical distribution per experiment to figure out what kind of scores they typically produce. The purpose of this is that in later searches, we can standardize our scores to this expectation and generate sort of reliable scores for our corpus. Once we have these standardized scores, we then weigh them. So we weigh the gene components by some experiment metrics. And we do this because in practice, we found that despite correcting for some nuisance factors in the first step, experiments still get scores that are somewhat correlated with some confounding variables, such as sort of how many differentially expressed genes an experiment has, the quality of the experiment, i.e. whether it has a batch confounder, et cetera, and some other features that are sort of non-biological signal in the corpus. Together, these two steps aim to create a reliable per experiment per gene score that roughly answers the question, how related is each gene to the contrast studied in a particular experiment? The last and final step that we take with these experiments gene scores is to aggregate them by a statistical test to yield a metric per gene per contrast now, rather than per experiment. And this is meant to describe how significantly each gene contributes to a contrast by looking across all the experiments in our corpus. To be sort of precise, what results is a set of contrasts, like you saw on the previous slide, that are ranked according to how much and how specific the set of query genes is differentially expressed. So that query list is the hit list of genes that we're interested in. This figure here is a summary of what I just described, but instead of stressing the process again, what I want to do is stress the evaluation of the algorithm. Obviously, given that this is real data, there is a relative lack of ground truth. We don't precisely know which genes are involved in various changes in phenotypes. And so while we did evaluate the algorithm on some instances where there are known involvements, such as sex-specific genes or genes that are involved in a well-defined pathway, a lot of our measures of performance are based on simulations. And so we developed a pretty extensive simulation framework, which includes both spike-ins, where we insert artificial data to the otherwise real corpus. And so these artificial orange data sets here are inserted at various levels, either sort of a few experiments or a lot of experiments. And we attempt to recover their contrasts as the top results in our search. The other kind of artificial data we do evaluation on is an entirely artificial corpus, where instead of having any real data, we actually model each gene's specificity to each contrast. And so we have a fully characterized data set where we know which genes should be related to which conditions, and we can exactly characterize what's going on with the algorithm. Together, these kinds of simulations set our expectations about how well the method should perform on real data. And to sort of give you the general conclusions of the evaluation, we found that on one hand, the best, most related contrast in the entirely artificial data would rise to top rankings in the majority of the thousands of searches that we did. And more importantly, we characterize the times that it failed as times where there are very few instances of a contrast in the corpus. On the other hand, the rankings were found to not be random, but rather ordered according to their true specificity that we modeled. We also compared our algorithm to some previous art and literature, namely the Gene Expression Atlas's gene set enrichment algorithm, and one other one called Gene Chaser, which was published a few years ago. In general, we find that our algorithm outperforms the other metrics and can also perform in cases where things like Gene Chaser just can't work because of assumptions they make in the binarization. In terms of evaluation on real data, I'll just show you one example, and that is for a published gene set from a GWAS study for schizophrenia. And so we took 322 genes that were reported in this here study, which is a large schizophrenia GWAS. And we found that our method correctly identifies schizophrenia here, as well as a host of other neurodegenerative diseases as the top rankings. And so you can see various cognitive disorders, mood disorders, schizophrenia, and bipolar disorder showing up here. Furthermore, and I glossed over details for sake of time, we found that using our method, one could prioritize the original hit list of 322 into a smaller subset of, roughly 132, so this number over here, genes that are frequently and robustly differentially expressed according to our corpus. And so instead of having to examine all 322 genes, a researcher could narrow that down to about a third of those using our method. Of course, it's not perfect. We're largely at the mercy of the amount and the quality of our data. As you can see here, we're at this line, and this shows the progression of geo over time. So we have a long ways to go to catch up to what's available in public repositories. But as we continue to gather more data and get more familiar with the nuances in this kind of analysis, we expect that we will become more robust and offer even more meaningful insights for gene function prediction. With that, I would like to thank my lab, especially Nathaniel Lim, whose preliminary work on this kind of analysis served as a starting point for my work. I'd also like to thank our funding sources, yeah, if there's any questions, I'm happy to address them in the live Q&A. Thank you very much.